Welcome to Hope Bio. We are excited to talk about traumatic brain injury and the clinical trial that is enrolling at UTHealth. So a good place to start is to talk about the intervention. What's the treatment? This trial is going to be testing the safety and efficacy of intravenously, so through your bloodstream, applied autologous, your own, mesenchymal stem cells for the treatment of traumatic brain injury. Okay, so the first step of this trial will require a screening visit. So visits 1 and 2 are your screening visit along with your fat extraction. So if you screen and you pass, you'll be able to go and get your fat extracted. Now, what does that mean? A surgeon will take a small amount of fat from anywhere you have fat or anywhere you request. They will put in some local numbing and then remove approximately 1 tablespoon of fat tissue. Now, why are we using fat tissue? Fat tissue contains the medicine that we are looking for. Okay, fat is a great source for a particular stem cell called the mesenchymal stem cell. Now, these are readily found all over your body, so you already have them. So what we're giving back to you is exactly the same as what is already running through your body. But these cells do a variety of different functions, regenerative functions. That's why we're studying them for traumatic brain injury. But these cells are limited in number. And so the idea is we want to increase the number of them and give you them back in larger doses. So how many will we find in a small amount of fat? Not too many. Approximately 100,000 cells is what we'll find in an average person. So this is a culture flask, and when cells are put into a flask like this, picture it like a petri dish, the cells will adhere to the flask. That's because they're actually plastic adherent. They like plastic, and so they stick, and then they'll begin to proliferate and grow and spread across the flask surface. This is what we do day in and day out. We turn a small amount of cells into a large amount of cells. And so it goes through repeated generations. So we'll take some of these cells and seed more flasks to get more cells. So it will take us about 6 to 8 weeks to create enough cells to create what's called a master cell bank. We will take the cells we've been culturing from that original fat. So we're gonna package these up into little cryovials like this, and then we're gonna store them in really, really cold temperatures so that they're like dinosaur age frozen. So that's what happens during the break time. We go through the banking process, and once you're banked, your cells, your original MSCs are banked. From there, just like a yeast starter, we are able to make subsequent treatments, about 1,000 of them actually, with those stored cells. So in this clinical trial, there will be 3 administrations of cells that will occur every 2 weeks. So your coordinator will send you a schedule and you'll receive those dates for 3 visits that'll happen 2 weeks apart. And we're packaging up 200 million live cells. How do we know they're live? So after we've packaged them, we have a small sample that goes to quality control that will test these cells in accordance to all of the criteria that are required to release a product. Once those cells have passed testing, they will officially be released for packaging and delivery. So that's the whole process. Once the cells are delivered to the clinical trial research site, when you, the participant, comes to check in, they will put an IV in, and the cells will be dripped over a 1-hour period. There's also an observation period post-infusion to ensure safety. So I think now is probably a good time, since you know everything about the product, to go find out more about the details of the trial and go see if we can track down Dr. Cox. It's carpool karaoke time! Just kidding. We are on our way to the Texas Medical Center. Happens to be about 20 minutes away from our lab. So once the products are made, they'll be hand delivered, hand couriered to the Texas Medical Center. Some fun facts about the Med Center.
They have over 750,000 ER visitors each year.
That is a lot. They have about 3,300 visits per day. These are patient visits per day. That is a lot. And it is the world's largest life science destination.
And the parking's terrible.
Okay, driving and parking may not necessarily be the most fun, but that's what Uber is for. And you can't compromise on the best care. And don't hold me to it, but I do think that in this trial there is a parking stipend as well. So that makes it good. It is a beautiful drive. So we are here at the McGovern Medical School at UTHealth, and we will see if we can catch Dr. Charles Cox. Dr. Cox is the principal investigator for our TBI trial. He is professor of pediatric surgery and neurosurgery here at UTHealth. He is world-renowned for his work in innovative treatments for conditions like traumatic brain injury, and particularly with this innovative use of stem cells. So he's definitely well qualified and the guy that is in charge of this entire trial and really the creation of the protocol that we're here to discuss today. So let's see if he's here. He is festive. Hi, Dr. Cox.
Hello, Dawn.
Want to answer some questions about TBI?
Waiting
Oh,
to
okay. Well, thank you, Dr. Cox, for making time for us. We are going to just go over the basic structure of the trial. But first, could you give us a brief explanation as to why? It was because we did a TBI-01 trial, which was the first TBI trial, which is a phase 1/2A trial. And now we are embarking on a TBI-02, the second phase 2. So why? Why are we doing another trial?
The why, in short order, is because we're very encouraged by the phase 1 results. And so the idea being that the infusion of cells over a period of time, over 6 weeks, 3 doses, was going to be impactful in terms of for patients, their neurocognitive outcomes, how they feel and function. And then for us in the science piece of it, that the imaging parameters that measure the body's inflammatory response to this injury chronically is downregulated. And so we saw that in the phase 1 trial, and recognizing that there's limitations to the phase 1 trial, meaning everyone gets treated, everyone knows they got treated, and so that means that there's some baked into it, some bias. But that wasn't the purpose of the phase 1 trial, and it was just to establish or reestablish, confirm safety, and then determine the treatment effect size. And so we were able to determine that allowed us to plan the phase 2 trial with the appropriate number of patients, so that when we now are moving on to a blinded trial where people don't know if they got it or not, and it's randomized, and all the people involved don't know, that our ability to detect a difference is robust enough in terms of the number of patients.
So, for those that don't really understand anything about stem cells, can you describe what the cells are doing?
Yeah. So, way back 20-something years ago when we started doing this, there was a idea that engraftment of cells, meaning you're doing a transplant, functionally taking cells, you infuse them, and that they take up residence in the brain, in our case, and that they, in very basic terms, if you had a normal cell and an injured one, it would come in and kind of bridge that gap and kind of reestablish connections, et cetera. I think now most of us understand that that's not what's going on. And really what we think is occurring is that the infusion of these cells alters the inflammatory response to injury. It doesn't get rid of it completely, it down-regulates it, such that the endogenous repair responses in the body are allowed to occur, and that damaged but not dead neural pathways are able to come back online over time. And so we measure that with these very specific tracers that are used for that label activated immune cells in the brain. And then we measure microstructural changes in the brain using a very specific type of MRI, not the type of MRI you get when you go in clinically for, if you're getting some evaluation of your brain or spinal cord or something like that. It's not that type of MRI. It's a bit more advanced type of MRI that looks at very specific fiber tracts and then these other metrics of how the cells are in those specific regions of interest are functioning.
So where is this trial taking place?
So Houston and San Antonio are the two sites. So UT Health Science Center San Antonio is our secondary site that we've brought on. The part of the point of doing that is as you start to move a technique or technology or something out to different places, it improves what we call in clinical trial lingo the generalizability of it, meaning it can not only be done here, but it can be done somewhere else. It's exportable, and that's important, obviously. So you can treat people wherever they are, as opposed to everybody coming to one spot. It also speaks to the biological effect of the intervention, meaning if there's a true effect, it's going to be reproduced somewhere else too.
So I understand San Antonio is not recruiting yet, and Houston is. Do you have an anticipated date as to when San Antonio?
Yep, San Antonio is coming online a little bit later because they're developing the tracer technology that we already have and are being used, and so that takes a few months to come online. So the hope is probably in another 2 months or so that we'll be able to start enrolling in San Antonio.
And so for everyone viewing, we are going to put in the link in the caption below, we will put updates as to when sites are live, and if recruitment is full, we'll provide all those updates below. All right, so let's talk about the trial design. How many visits? What's the commitment?
So it is a big commitment. And so the trial has numerous visits, obviously each of the infusion visits, and then there's a baseline visit, and then there's follow-up visits. And so that all takes place over a 2-year window. So it's a thing. It's not just, I'm going to drive in, I'll get an x-ray today and get my infusion, and then I'm done, phone call a little bit later. It's a much more extensive commitment. So there's the screening that goes on, and you have to gather all medical records and make sure that things meet the enrollment criteria and none of the exclusion criteria. Then there's a baseline visit that has examination, lab work, imaging, and then fat tissue harvest, cell isolation, and then there's an interval for the cell isolation and the expansion. So it takes a little while to expand the cells. So that's a 6 to 8 week period. Then you come back and there's infusion, infusion, infusion. So 3 infusions, 2 week interval between the infusions. Then we have 6 months follow-up, repeat imaging, and then follow-up in a year and at 2 years with just either a video or phone visit. And so I've had the MRI protocol myself, so that's not too onerous. But it's a sequence of those imaging studies that you have to lay flat for a while, and the whole process on the imaging days is about a day-long event. And then if you don't live in the area, there's travel on the front end and the back end to come for this and for each of the infusions. So there's a commitment in terms of time, effort, and travel.
Can you describe the imaging?
So the imaging is a combination of what's called a PET scan, positron emission tomography, and a MRI scan. And so the MRI is where you lay in a tube and you have to lay still, and it takes about 30, 45 minutes to get the images. It's loud. It's painless. If you're claustrophobic, it can be a little bit aggravating, but I don't think it's too much to I think everybody, if you can do it, you just close your eyes, you get to pick the music you want to listen to. So it's not too bad. The PET scan, you're in a similar scanner, but it's not as claustrophobic, but they give you an injection of the radio tracer, which binds to the immune cells in your brain and then is cleared out of your body rather quickly. So quickly that they have to make a dose of it because it's going away so rapidly, it's decaying so rapidly, so they have to give you the dose, get the image, and then we look the PET scan and the MRI and merge those two things so that we can tell exactly where in the brain the immune cells are activated. And so that's why that's the two of them together.
So we would say 8 physical visits and 1 phone call. So the 8 physical visits would happen during a year's time. And if people are traveling in or are in Houston, the first 2 visits, which are the screening and the consent, and then the baseline imaging, would happen together. So you could travel once, come in once and get both done, and then travel out. And then you've got 3 visits, and then the next at 6 months, you can travel in and do both of those.
Yep.
Right. And so after your 3 treatments, you get the imaging done 6 months later, and then 1 year later, you come for a final exam, and that's essentially the physical commitment that needs to be made. All right. Let's talk about who are the candidates that you are looking for. Inclusion criteria are the criteria that are required to enter the study. This is what you need to be or have. Okay. 1, adults between the ages of 18 and 55. Documented functional neurological damage to the central nervous system from closed head trauma that is unlikely to improve with present standard of care.
Right. So that means whatever you've done, you've hit a plateau, so you're no longer improving, so that we know that the point of that is we don't want, if someone's getting better on their own, we don't want that to be what's attributed to a treatment if they're just getting better on their own. So it's you plateaued in terms of your outcomes. You have to have an impairment on this scale, the Glasgow outcome scale. So you can't be back to normal, even though you had an injury and you're back functioning and doing things and have very minimal symptoms. We want patients who are impaired enough to where we can see a benefit from the treatment. If you've already recovered, then we don't want to enroll that type of patient. But it can't be so impaired that they can't interact with any of the neurocognitive testing.
So then with the Glasgow outcome scale that's greater than 2, less than 6, can you tell us like symptomatically where a patient
someone who's not vegetative, so the Glasgow outcome score, the 2 lowest are dead, can't be enrolled, vegetative, and then there's severe impairments, it has 2 subcategories, moderate, and then good outcome, which has, and each of those have 2 kind of high and low range. So you can't be on the highest part of the good, and you can't be on the dead or vegetative scale. So it's the patients who are in between. What that means functionally is patients who still have some type of deficit that keeps them from working, require on the higher end of the scale, and as you start to migrate down, it's what degree of assistance that you need for your daily activities, all the way to pretty much constant supervision on the lowest end of the scale.
Dr. Cox, one thing we did not talk about was, we talked about the qualification of the severity of the TBI, but there are many different kinds of TBI. What TBI are we specifically looking for in this trial?
So, well, we're agnostic to the, there's extraaxial hemorrhage, there's diffuse axonal injury, there's all of these imaging related classifications of TBI, and we're agnostic to that. But we do exclude penetrating traumatic brain injury, so a gunshot wound to the head or a other type of penetrating wound to the head is an exclusion. A host of reasons for that, but one of the big reasons for it is it almost always invalidates the MRI because there's metal fragments, et cetera, within the brain, which makes the imaging impractical. But we're not, we're fine with enrolling patients who've had blast induced TBI versus a contusional TBI.
How about hypoxic?
Well, so hypoxia isn't traumatic. It's also often a component of TBI, meaning there's a brain injury and then there's a period of time where you weren't breathing, which is additive to, and that occurs with a lot of patients, and it's usually reflected in their level of consciousness. So what got them enrolled into or classified as a severe TBI to begin with.
How about conditions like cerebral palsy?
That would be one of the things that would be a exclusion criteria, and in terms of the functional deficits. And again, not because there's any real biological issue with that. It's that it's going to change some of the functional outcome metrics.
Onset or diagnosis of the injury greater than 6 months. Ability to obtain consent, and the ability to communicate in English or Spanish. Very straightforward. Okay, so exclusion criteria. Now, this is the list of criteria that one cannot have, and if that would exclude you from participation. So 1, known history of, and there's a bunch of things here, but I would say all in all, it has to do with the criteria is written in a way where we're really trying to exclude anyone that has a significant comorbidity that would conflict with the trial. So intellectual deficiency or psychiatric conditions likely to invalidate the ability to assess changes in cognition or behavior. Recently treated infection. Well, we get that one a lot. How soon? That's
a judgment thing in terms of severity, what the organism is, what's the, and so if a person has something as extreme as tuberculosis that's required extensive treatment and they just finished, that would be something that we would be concerned about reinitiating a therapy that has an immunomodulatory impact. If you had a strep throat 3 weeks ago, we're okay. moving forward. So there's a wide range of things, and we couldn't write those criteria such that it incorporated every possible circumstance. So it's really up to our judgment at that point.
Renal disease or altered renal function, and we are basing that off of blood markers. So blood tests will be performed at screening, and in this case, an EGFR less than 60 would disqualify you. Hepatic disease or altered liver function, SGPT greater than 150, cancer, any history.
Yeah, so, and again, that some of those things are, if you had a small basal cell cancer right here that was excised by your dermatologist a year ago, we're fine with that. If you had stage 4 breast cancer and even though you're cured, we would be, or in remission, you've had, we would be reluctant to treat somebody on that because of the potential immunomodulatory impact of the. So again, some of those things are judgment calls based upon our assessment of the situation.
Immunosuppression.
So someone who's had another organ transplant or have severe dermatologic diseases, for example, that psoriatic arthritis, for example, that may be on significant immunosuppression. While this therapeutic may be helpful for that condition, paradoxically, we don't want to use that. We don't want to be using it in conjunction with something where we don't know what those interactions would be yet.
HIV, in addition to HIV, it's not written on the exclusion, but this trial is studying the use of one's own cells. And in our process, in order to qualify for banking of the cells, which you need to bank in order for us to be able to make the treatment, one must be negative for HIV, hepatitis B, hepatitis C, and syphilis. So I would add those to the list. Chemical or alcohol dependency.
Yeah, because those impact your neurocognitive outcome. And so those are confounders. If you're, so those would be impactful for our outcome measures.
If one said a glass of wine. Right?
Well, then I would be excluded. But so I think dependency, the way we measure dependency is, it depends a little bit on what the substance is, but screening positive or requiring a rehabilitation process to re-engage in your activities of daily living, I think are the issues.
Acute or chronic lung disease requiring significant medication, bleeding disorders, including immune-mediated heparin-induced thrombocytopenia, known sensitivity to heparin, Lovenox, and pork products. So in this trial, Lovenox is also being prescribed, and so you can't have a sensitivity to that. Dr. Cox, why pork products?
That's where it's made from. It's made from porcine intestine.
Individuals with mechanical prosthetic heart valves, individuals who have received a stem cell treatment.
Right, so we don't want another therapeutic confounding our intervention, right? So if you've, if you've been on another trial and then starting this one, I think would just be a confound. It's not so much that we're medically concerned about it, it's just a confounder for the treatment.
Normal brain, CT, MRI, spinal deformity, spinal surgery, complete spinal cord injury.
And that's because it would impact our functional outcome measures, but also then the hardware can also impact our DT MRI.
Diagnosed with a genetic or metabolic disorder related to the neurological condition. Other acute or chronic medical conditions that, in the opinion of the investigator, may increase risks associated with study participation. Some examples?
So if you have a hereditary thrombophilia and you're prone to developing venous thromboembolic disease, I would say that we should avoid that type of patient.
For women of childbearing potential, positive pregnancy tests at screening or visit. For this question, I was wondering, for a participant that is, say, not pregnant or not planning to get pregnant immediately, but of course would eventually, do you foresee any risks?
No, I don't think so. For this autologous strategy, there's no alloimmunization or something like that.
Okay. Concurrent participation in another study. So we don't.
Right.
Yeah. Inability to undergo the diagnostic tests, like the PET imaging and the DTI MRI, or unable to cooperate with any tests or outcome assessments. And the last one, unwilling or unable to return for the follow-up visits.
Right. So it doesn't benefit anybody if we don't know what the effect of the treatment was. And the only way we can know that is you got to come back.
Correct. Okay, so this trial has a placebo arm. So that means, and is it 50-50?
No, you're more likely to get the treatment than not. So, it's not the coin flip of whether you get it or not. So it's got a weighted to be more likely to receive a treatment than not. And then at the conclusion of that, once everything is to the end, then you'd have the opportunity to get infusion if you were on the placebo arm after we finished.
And this is after all the data is in and we have seen, obviously we wouldn't invite the placebo people back for a trial if it didn't work.
If it didn't work, or there was some subpopulation in which it was higher risk or something, that exactly. So if all the data's in and it looks like there's a benefit, then yes, then you would be offered that as a therapeutic at the end of the 2-year window.
And there's lots of also contingencies to that. I mean, we would have to also make sure that find out where we can do the trial. It may not necessarily be here.
Yeah, there'd be a bunch of, yes, there'll be a lot of
what ifs, but we will do everything possible. If the trial shows that the intervention does work, we as a study sponsor will try to do everything we can to make it available to the placebo group. Risks, can you tell us about risks of
so
cells?
There's a theoretical risk of any type of infusion. Either there's an undetected contaminant, we've never had that happen, reactions to any of the cell preservatives that even if they've been washed off, some people could have a hypersensitivity reaction. We haven't had that. And then there's a theoretical risk of developing blood clots from the infusion. That's why we give the Lovenox, and we haven't seen that as a problem either. So most of the risks to date are theoretical and that haven't been borne out in the patients that we've treated or have been treated in other protocols with the same type of cell approach. So I think it's a safe, low risk.
Are there any costs involved in the study?
The cost to the patient are just regarding travel and the around that, but the patients aren't charged anything. The federal grant pays for all of that, for the cell manufacturing and the imaging and the neurocognitive testing, et cetera. You just have to get here.
Who's funding the study?
So your tax dollars at work is the Department of Defense through something called the Congressionally Directed Medical Research Program, CDMRP. So
what is the most important thing you would tell anyone interested in participating?
That it's a commitment. It's you got to commit to doing this for this period of time, the people that we studied in the Phase I trial, 24 patients, the overwhelming majority of them were very committed to it, and I think part of their commitment was enhanced by the fact that a number of them had significant improvements in things that they could recognize, not just our imaging studies or the lab work, but things that they would come back and report, some things that we couldn't have predicted. And so I think that that's part of what reinforced their initial enthusiasm.
Their participation, your participation, will lead to this body of data that hopefully can support this treatment going forward.
Yeah, so that's the idea, is that this would get us to the next step, and that we could then evaluate this in a pivotal trial that allows us to kind of move it to an established therapeutic.
Again, all the information about the trial will be linked in the caption below. Details, there's a clinicaltrials.gov link which lists out all of the inclusion, exclusion, and also the contact information for the trial sites, both trial sites. Please, please, please watch this video in the entirety to get all the information before you call in, but we're always available to answer questions. Dr. Cox, anything else you want to add?
I think we got it.